Okay, so hello. Could you introduce yourself to our audience?
Yeah, absolutely. My name is Rebecca Vaught, and I am the founder and head of Van Heron Labs. Van Heron Labs is a bioplatform, and what we do is we harness multiomics data in order to determine custom nutrient profiles for cells. So in a practical sense, that means that we make media formulations and growth and expression feeds for various types of cells and cell lines.
So yes, why is this an important sort of technology? Why is this an important platform to provide?
Yeah, good question. So the first response would be that we think it's really important to use genetic information more or less as a tool. So everyone up until this point, a lot of folks have used genomic information. It's kind of been sequencing for the sake of sequencing. You know, we have some really interesting new microbial strain, new plant species. Let's sequence it and see what we can find out. And that has sort of been the story, honestly, for the past 20 years. And I would say the story for the past, really the past 5 years has been, you know, what can we interpret from that genomic data? What can it actually tell us that is important that we can utilize in order to enhance biology, and then in that pursuit, make the world a better place in some way. So we founded the company because we thought the platform could be deployed very much in that way, and that it was important to be able to harness genetic information in this way. And then the way that we're deploying it, or kind of the vehicle that we operate in, is this culture media realm. And within that, so you guys are very focused on you know, the synthetic biology space. So in that space, of course, everyone is very focused on the cell lines. They're very focused on the genetic engineering techniques, whether it's CRISPR, you know, trying to put those particular pathways in, you know, switching things on and off. And then a lot of attention is being paid on the product itself. You know, is, you know, a brand new type of lipid biomaterial that does not exist in nature that can be used for some particular purpose, or a biologic drug, COVID vaccines. You know, the examples go on and on and on, and we're just going to see more and more of this. Don't want to preach to the choir because I think your audience knows the importance, but one area that's been very neglected or overlooked is the nutrient environments, or just the broader context that those cell lines and products are growing in, being propagated in. And we think that this is very, very important. So it's one thing if you can engineer some new product that is going to change the world, but if you can't scale that up and make a biomanufacturing process around it and produce that product in a way where it will be commercially viable, you'll be sitting on something like biofuels, which has all this promise and could change the world, but we can't wrap biomanufacturing around it. And so the whole field essentially failed. And so we don't want to see that happen with synthetic biology, and with biologic drugs. You know, insulin is very inaccessible for a lot of patients that are out there. So we think a lot of times, COVID is a great example. What if you can produce much more of this biologic drug from the same amount of infrastructure, just by changing the culture media? You know, you don't have to further engineer your strains. You don't have to buy a new type of bioreactor. All you do is just change the nutrient context that's there. And so there's very few companies that are paying attention to, there's very few companies that are even making culture media. And there's even fewer companies that are doing it in any sort of like custom context. And we think that we're really like the best solution for folks, because instead of making physical products, what we're doing is actually licensing formulations or giving people recommendations. And so not only are we optimizing the nutrient formulations, the media formulations, but we're really democratizing the industry, because we're giving those tools back to the companies that are producing those strains and products, and saying, this is what you need, and this is the recipe that you need to follow to be able to make it, so that they can make these massive bioprocesses and scale them up. And so we think this is really critical, and we really have been rolling with this idea over the past couple of years that we want to be the foundation of this bioeconomy that we're living in. So when you have some great strain that you're working with or some life-changing biological molecule, we hope that you partner with us and that we're able to enable you in that way and get that technology out into the world by pairing it with culture media that's going to be successful for scaling everything up and producing that product.
So essentially creating a platform to scale biological solutions through the sort of, in some ways, the individualization of media for different bacterial strains or strains of, other organisms.
Yep, that's right. So we work across bacteria and then also with cell lines. And then a lot of what I've been focused on is kind of that biomanufacturing perspective. But we also, one of the big places where this is needed is for research where they're working with something that is hard to mimic those in vitro conditions to some sort of in vivo state. So something like tumor biology. So if you want to test something like cancer therapeutics, you need an in vitro environment that's going to match the in vivo conditions of interest. And so in that case, you sort of need this precision nutrition approach where you actually look at the genomic information and then design that environment around it. So our clients that we work with are kind of on both sides of that token. So on the biomanufacturing side, and then kind of more on the research side, where you need this very high powered nutrient environment to match whatever it is that you're doing.
So in the stead of sort of, I'm so sorry, I'm going to get my words right. So currently, how is media designed and optimized? Sort of, obviously, you are pioneering a new sort of using wide genomic data in order to produce this platform for optimization. But how is most media designed and sort of used and produced?
Yeah. Good question. So up until this point, it's been a lot of trial and error. And then the next generation was a lot of high throughput methods of still also trial and error. So microfluidic devices, robotics platforms that can grow identical cells and then change the culture media formulations by one component across that array of cells, so that you can look at, depending on tweaking individual ingredients, what the cells respond best to. And then from there, you take whatever the formulation that the cells responded best to, and that becomes your media formulation for your bioprocess, or they bottle that up and sell it as something like a chemically defined media. So those medias are actually, it's still a good approach if you can access that. The problem a lot of times is cost. So those methods were developed for the biopharmaceutical space, which we do work within that space, but we also want to kind of democratize this. And so just engaging with one of those companies that deploys one of those platforms, it costs millions of dollars. And there's a good chance that they're not going to let you own that IP. And so there's issues. And then a lot of times what our platform actually tells us are novel ingredients that have never existed within a cell culture setting before. And so if you're just playing around with the same 60 ingredients in kind of this iterative way, you're missing a lot of the novel nutrient picture that we can pick up with our process.
And could you perhaps explain how do you use the omics, the multiomics approach to further develop the culture medias for specific strains and cells?
Yep, I definitely can explain that. And I also did just want to add on to the last point that folks also use mass spec to get at this problem. So you can put in a sample of the culture media, and then that can tell you the macronutrients or the micronutrients that are there. The problem is that you have to run two different processes to get those two things, which again kind of comes down to that cost and the time and the accessibility. And then when it comes to the multiomics, so yeah, multiomics just means multiple genomes. And the word, depending on the person who's using it, it actually means multiple things. So one of the context is that it can mean multiple genomes. So for a plant species, you actually have 3 genomes that are present. So you have the nuclear genome, and then you have 2 organellar genomes that you have to worry about. So in that sense, taking a multiomics approach to plant genomics means capturing all of that genomic information and getting a complete biological picture. Multiomics also means getting multiple types of genomic information. So that could be a genome, a transcriptome, a proteome, a metabolome. And so, even there's a million types of omics data out there now. And so for us, it just means we use both, in both senses of the word, we use combinations of those things in order to get that complete biological picture, which I think is what really sets us apart. I'm very fortunate that I work with a bioinformatician that is from kind of the evolutionary and invertebrate biology side of things. And so they have to look at everything because they're working blind. There's no model systems for a lot of those organisms. And so they were capturing the lab that I happened to do my undergraduate research in. We were capturing everything that was there and doing de novo sequencing back when it wasn't cool and it cost a lot of money. And now we deploy that regardless of this situation because we want to look at everything that's there.
That's it's actually it would be interesting to know, how did you sort of decide to form a team and work on this problem? Sort of where did the sort of inspiration come from? How did you decide, yes, let's start up,
Yeah, good question. So if you looked at my background, it doesn't have anything to do with cell culture or media development. I did my PhD in evolutionary genetics. And so me starting a company doing this is very unlikely, I would say, as me as a founder. But there are some ties into the core platform and kind of my upbringing as a scientist. So my bachelor's degree had a lot of physiology and biochemistry incorporated within it. And then for my PhD, and then I was doing undergraduate research in evolutionary genetics and kind of working with these unusual invertebrate organisms in these unusual environments. And then juxtapose that with a bachelor's degree in molecular cell and microbial biology. And then for my PhD, I switched gears again and started studying evolutionary genetics, but in a different context. So kind of thinking about really big picture questions about how the nuclear genome and the mitochondrial genome talk to each other and what that means. Specifically, the first part of my PhD was all focused on what those interactions and what each of those genomes means for aging outcomes and performance outcomes for organisms. And in the last year of my PhD, I started to get really frustrated because a lot of folks that were working in the nutrition space were able to drastically increase and decrease lifespan. It felt like they could just switch it. They could just decrease lifespan by 50% and increase lifespan by 50% just by tweaking these macro and micronutrient concentrations. And so I just got really fascinated with it from that perspective. I'm just like, it almost erases what the genotype is. It's this nutrient environmental context that has this bigger role for the organism than its chromosomes. And I was really, I had lines that had X chromosomes pulled in and out and mitochondrial genomes pulled in and out. And then somebody over here who added sugar to their media is like totally beating me on playing with aging outcomes. And so I just got really fascinated with it from that perspective. And just kind of got exposed to or started thinking about how you can use genomic information to make these inferences on the nutritional context that's going to be most appropriate for a cell or an organism. And from my bachelor's degree in microbiology, I knew how outdated culture media was. And so that was like when I put those two things together, basically, it was like, oh, well, this is going to solve so many different kinds of problems. And then that was kind of my first thought was like, oh, this is what I should pursue next. And then the second thought was that I'm going to have to form a company around this. Like, this really can't be, at that time I was still thinking about postdocing. And I was like, this really can't be a postdoc because I need to work with bacteria. I need to work with other organisms, kind of across the board and prove this out, that it's a ubiquitous platform. And so that was kind of the reasoning behind the initial idea for Van Heron Labs. And then got paired up with my co-founder. Obviously, I didn't have a big biotech network. I was living overseas in Australia at the time, working evolutionary genetics, so I didn't know anybody who was producing vaccines or working within the biotech space, really, except for some of my friends that I went to college with. And they were in the Texas Medical Center. And so that's how I came back to the US and went to the TMC and started the company and reunited with my co-founder, my now co-founder, Alec. He had been doing, he was very immersed in kind of biotech and life science entrepreneurship, and had been doing consulting with biotech companies. And I was like, yeah, I need to go to Houston and see what my friends are up to and see if they can help me get this off the ground, which is what they did. So, and then ever since January 2020, that's what we've been doing.
One of the things I'm interested, sort of going back to sort of the fact that other approaches, the current approaches you were talking about, such as the microfluidics and I believe it was the robotics approach of making identical, was it like cell cultures or identical cells? So how does your approach reduce costs? Because I'm not quite sure if I quite understood from what we were talking about before.
Right. So yeah, it's just that those people are that have those micro robotics platforms, they've spent millions on that, and they're just trying to recoup their value. So they charge a lot of money for you to access it. But in a biomanufacturing context, the way that we think about reducing costs is in this vein of what if you can produce a larger amount of biological products, so substantially more yield, which is ultimately what you're selling, with the same amount of infrastructure. So from a 100 liter bioreactor, you're able to get 100 grams of enzyme. What if we can double that? What if we can triple it? That means that you can sell 3x more of that enzyme. And so we think about reducing costs in that way. So it's really kind of a margin increase for their business model. And then the other thing that opens up for companies that are kind of in this biologic space on the biopharma side is, okay, with something like insulin, if we 3x their manufacturing, they could still produce, so they could produce way more insulin. They can make a lot more money on it and also reduce the cost of insulin. So, and that's kind of what we need also on the synthetic biology side is a lot of these things are not economically, like the unit economics are off, right? So yeah, they may have some disruptive, biomaterial, but the cost to make that is 100x what it costs to make a petroleum product. And so we can help get folks closer to that to price parity through increasing the output of their biomanufacturing. So that's usually what we're thinking about when we say lower costs. However, a lot of times what we find is that we can actually lower costs with the ingredients themselves. So for some folks, just switching them from pharmaceutical grade nutrients over to something like food grade can also go a long way. And so for folks where it's like pennies on the dollar, we can fix it.
And could you explain what's the difference between pharmaceutical and food grade? What does that mean?
Yeah, good question. It usually has to do a lot with the purity of the product. So for pharmaceutical grade, it's CGMP is the certification. So their process has to be overseen and approved by some sort of governing or regulatory body. And usually like the actual difference in the nutrients themselves is the level of purity. So we find sometimes with the pharmaceutical grade components that there are contaminants within them, and those contaminants can do funny things like cause reactions and lower the pH values. But if you have some sort of really robust process, for instance, for a lot of folks that are working with microbes, the ingredients at the end of the day may not make as huge of a difference. And so they can switch over from one to the other. But for other folks, they have to stay within those pharma grade margins. And the cost difference between the pharma grade ingredients and the food grade can be anywhere from 10x to something like 100x. It just depends on the nutrient.
Right.
Mhm.
And you mentioned that when you first got into the market or the industry of like cell media preparation, there were only like a few companies that sell those media products to labs, for instance. And we're just wondering if like, was it hard to start like a company that developed like cell media when the market was basically centered on a few companies that everyone knew of?
Yeah, I mean, I think our value proposition is a bit different. So, and I will just say there's a lot of companies that are making culture media. If you go out there and Google it, but usually they're specific to one or two cell types. There's only a few companies that are making kind of across the board, doing like all different kinds of cell lines. There's just a few huge companies that are out there. And so I think our value proposition is very different because we don't ever make physical products. We're always handing off a formulation. And so for some folks, you know, there are pancreatic tumor medias out there, you know, and if $1,000 a liter is not a big deal for you, if your company's worth several $billion, then you don't have any reason to switch to our media. Especially if you don't need to, like we were saying, get more product out of your process. And so our solution is probably not like the lock and key solution for everyone. But I think a lot of folks are interested in this. And so we sit in a very unique place in the market where we're actually selling. Ultimately, what we sell at the end of the day is knowledge. So we're licensing a formulation to companies, or we're actually giving them recommendations on the bioprocess. And so I think the companies that are it is hard to convince people maybe to switch from an off-the-shelf product, or why would I, you know, license a formulation from you when I can just buy XYZ from you know, Thermo Fisher. But for some folks, you know, it's not viable for them. We work very closely with a company that's at our institute, and they have to make an enzyme right now, and they are the media that works best for their cell line costs $1,500 a liter. And so they cannot use that media. They can use it at a small scale, but once they're in those bioreactors, it is not economically going to work. And so those are the people that we find want to partner with us, and have us make custom solutions for them.
So one of your newest tools is Cerax. Could you talk a bit more about Cerax and what it does?
Yeah, we're very excited about this. So this is not, this Cerax doesn't have anything to do with our core platform. So it is a new service that we have designed to help our partners better eliminate serum from within their processes for those companies that are working with cell lines. So for your listeners that are unaware, serum is, it does have to do with blood serum. So it's a blood or an animal product. So it's either derived from animals or even humans. And so it's very heterogeneous. So there's lots of things within it. It's everything that's in kind of your bloodstream. So hormones, solutes, proteins, everything is just kind of floating around in there. And people have used it to culture cells for a very long time. And the reason why is because it's full of growth factors and all these lovely things that the cells just, when they get it, they grow like crazy, right? It kind of puts them into this fetal state. It gives them everything that they need. And it can close the gap if you are working with a crappy media formulation. So if you're working with something that's not ideal for your cells, if you put a bunch of growth factors into that situation, they're going to grow no matter what. And so there are a lot of downsides with using serum, which I can go through a little bit. And so this new service was designed to help our partners eliminate it. So for folks that are making, several of our clients are cell cultured meat companies. And so if you're trying to make a laboratory grown meat product, you can't have any animal serum, bovine or human, in your product. Nobody is going to eat it, and that defeats the whole purpose of what we're trying to do here. So those guys have to get rid of serum. And then kind of what I was introducing is that we've also been working with folks who are doing like cancer research, COVID research, that kind of thing. The thing about serum is because it's this heterogeneous mixture of hormones and solutes, it is going to drastically alter the metabolism of your cell line that you're working with, which is going to be a far deviation from whatever physiological state you are supposed to be working with. And so what we realized from working with clients, kind of deploying our core platform, is that a lot of their big headaches were actually serum issues. And that was really the problem that they needed to solve, was that they really needed to get rid of serum in order to enhance what they were doing. The other thing about our platform is, so it is only focused on macro and micronutrients, which are small molecules. And so we don't, our platform doesn't have anything to do with growth factors or proteins. And so whenever we deploy VHL within a culture context, we can drastically eliminate serum, but we can't fully eliminate it. And so I was just kind of watching my clients like struggle with this as their core issue. And we were like, had our hands tied behind our back, because it's like, we can only get you most of the way there. Like, we can't get you to fully eliminate it. And I spend a lot of time kind of just putting myself in my client's shoes and thinking, you know, what would I do if I was in their scenario? Like, what are the action items that we can take to get rid of serum? And so over the course of 2 years, basically working with the company, I had a folder on my laptop that just had, you know, anytime I had an idea for a step that needed to be taken to eliminate serum, or an idea for an infrastructure recommendation, or I found some new off-the-shelf product that we could actually reverse engineer the formulation that would help a client, I started compiling everything into this folder. And furthermore, kind of one of the things that VHL is good at is trying to find the driving variables within these chaotic biological systems. So you have the media formulation that has 60 components, you know, that's where we thrive, which is not where most people would thrive. And that's why there are so few people who on their LinkedIn headline, it says like media formulation specialist. And so, but in essence, serum is this kind of chaotic biological system. And so how do we tug on a lot of variables at once in order to eliminate it in this case? And so once I felt really confident that that folder that I had this system, like the system within it could be used to eliminate serum for any of our clients, that's when, well, actually that's not when, then my plan was to essentially get a big company to pay us for that. So keep that as a feather in our cap, keep it under wraps. And then when a huge company comes along, it was going to be but wait, there's more for them to work with us. You know, we've got this great platform to harness multiomics data. Oh yeah, and we can eliminate serum for you. And I started to get involved with, in particular, Curtis Firestone from Future Foods DAO, which is a decentralized autonomous organization that runs on the blockchain. And we were talking about what a travesty it is that knowledge is locked away within academic and then also industry silos. So companies essentially doing exactly what I was doing, which is like holding on to really important knowledge. Like, this is like the only thing that matters for cultured meat, right? This is what these companies, Memphis Meats is $1 billion company, and they only eliminated serum 3 months ago. So this is not an easy thing to do. And so I just felt like a hypocrite. I was like, yeah, Curtis, this is terrible. I'm glad you're founding Future Foods DAO to take care of this big problem. And then in the background, like knowing that I was kind of a contributor to this. So we did a 360 and or 180, I guess I should say, and made the service to sort of democratize access to this knowledge instead of saving it for some big partner. I mean, we can still give it to them, but we're also going to open it up to startups and make it financially feasible for them to access this knowledge. And then through actually the partnership with Future Foods DAO, eventually, this is several months down the line, we'll also be able to lower the prices for this even further, and also expand the number of people that they'll have a little bit of a watered down version of what we do. It won't be as custom, but they'll still be able to get the knowledge out there to the broader community. And so we're really excited about Cerax and the service, and we hope that we can help folks eliminate it, and it's a thing of the past.
Mm-hmm. How do you democratize a piece of knowledge? As in, how does that actually work in practice?
Yeah. Well, the first thing is to not patent it, and not stake any intellectual property on it. It is intellectual property because I came up with it, but like with VHL, folks are usually licensing the formulations from us. We do offer IP buyouts, but that's in the rare minority of cases. So the first thing you do, I guess, is just get it out to you make that available to anyone. So instead of you keeping it in your file drawer, you announce it to the world. So I guess that's the first way is not staking it and not patenting the system. And then the second way is by making access to that easier through collaborations with like what we're doing with decentralized autonomous organizations. So essentially they will have a version of Cerax that they can also deploy that can help get that knowledge out there to the broader public. Whereas the opposite side of this is where you would, what we were going to do is just keep it as a trade secret, and it was just going to stay on that folder on my computer. So that's the opposite scenario.
And then is that still profitable for you? Is it like you're licensing the intellectual property without sort of calling it yourself through this very arduous and long and expensive process?
You could, if you wanted to, intellectual property is very interesting. You can go on with this for hours. You could give people, in some scenarios, if it was really like, the place where intellectual property makes more sense is for things like a device, right? Where you physically invented something, and even if you don't make a patent on it, there always is some inventorship kind of tied to that. But for us, I'm not in those scenarios. If you wanted to get that knowledge out there, you could maybe do something like a royalty-free license, like where there's documentation that accompanies the knowledge transfer, the engineering blueprints transfer, whatever it is that says, hi, I am Dr. Rebecca Vaught, and I have invented XYZ. I'm giving you a royalty-free license for you to use this throughout the world with no further obligations to me, blah, 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 blah. And so you can do it that way if you so choose. We are not going to, I'm not doing any of that. I'm basically just going to make a PowerPoint presentation that has all the bullet points in there. And I'm not too worried about this. So I think companies, when they're very hung up on intellectual property, it shows them that they're not a very good company. Just since January, we have invented 3 new technologies to complement our core platform. So they're like further growth adjuvants. And so we're constantly inventing and innovating things all the time. And so we don't feel this need to like hang on to this as strongly. But your point about how are you getting paid for this? Obviously, I'm taking probably a financial hit, because if I was going to save this on my laptop, someone like, Pfizer or whoever is struggling with this, other media companies still struggle with this. They would be willing to pay a premium for them to have exclusive access to that knowledge, right? So so I am taking a bit of a financial hit for the greater good. But having said that, I am, right now we are charging fees for this, even though it's not what we would charge somebody like a Pfizer.
Right.
So there's a bit of a trade-off.
What do you think is one of the biggest challenges for democratizing like more information within the science, like the science field, basically?
That's a good question. A lot of it does come down to money, because people feel that they can be compensated if they really hold on to things. And we used to be like this when we were a small company. We used to really hold on to everything. I wouldn't even tell people much about my platform. I was very secretive and close guarded. And I think academics and people that have invented technologies are like this. And they feel this real need to be compensated, because what if they never come up with another great idea again? What if this is their life's work, is this one, device or something that they've invented? So there is a financial element to that. And then the other thing I think is showing academics that they can also become entrepreneurs, because the best way to get paid for your brain power is to actually start a company. And so I don't know if there's any academics that are listening to this, but I essentially am a PI in my own lab, which means that, I basically skipped a postdoc and now I run my own laboratory where we pursue whatever hypotheses I feel are interesting. The downside is that instead of grants, I have to bring in customers to pay the bills. So that's the difference, but I am able to have complete control over my intellectual property, and there's no university that can tell me what to do. And so I hope that in the coming years, we see a lot more scientists become CEOs. And that's a great way for technology to be realized out there into the world. And then there's a whole movement around this, right? DeSci, how do we decentralize science? How do we incentivize people, smart people, to work on big things? And so I think there's going to be a lot of conversations around the topics that you guys have just brought up over the next few years, and we're trying to be very active in that space as well. So I certainly hope that more technology does get out to the general public.
You mentioned the inventing 3 new technologies as of even this year, which is something I wasn't expecting because it's, I guess when I think of innovations, it doesn't, you tend to think of innovation as happening once, then you're like, okay, that was my moment, right? What were those three new inventions? And
yeah, absolutely.
so
I think
what were the aspects?
Yeah, I think for a lot of people, maybe innovation does only happen once, and that's why they're so guarded about it. But yeah, we get to do such fun stuff in the lab all the time. We're working on like hair care products behind the scenes and like all this crazy stuff, for things that we can devote little pots of money to in a little bit of time. And it's not a huge suck away from building the core platform. So that's a lot of fun. That's a fun aspect to my job. I hope one day that I'm able to actually put the CEO hat down and just be the CTO and be in charge of inventing the cool technology that the company kind of runs on. And working with teams who are also working on that. Definitely my lab team bolsters what we do, and they help talk me down from the ledge, and then also help me when I do come up with something that has legs. But yeah, so we have basically made another platform technology, and it's a few things. The word biosurfactant is what we kind of throw around, but it is really a plant-based. It can serve as a biosurfactant. It can serve as a nutrient delivery system. And so what we have basically figured out is a way to harness, or a methodology to make a reagent around this plant-based molecule that was traditionally very hard to utilize. We've kind of figured out the secret sauce on how you actually can make reagents from it. And so with that, we have made, using that underlying kind of technique or platform, ways to create growth adjuvants. So it also, it changes the fluid dynamics of the media in such a way that it increases the oxygen, the dissolved oxygen levels within the media. So using it can enhance the efficacy of VHL. And then also it very interestingly delivers VHL. So it delivers the nutrients to the cells themselves, so it can help them better uptake those nutrients, which is important. And then for expression, because it's enhancing the dissolved oxygen within the media, it can also enhance protein production. And then it can also deliver nucleic acids to the cells, so it can also be used as a transfection reagent. Yeah, so it's been a lot of fun to play around with that. We have a research fellow So he was an intern for the past 2 semesters, and now he is graduated, and he's going to be a research fellow for us in the lab. He is one heck of a chemist, and so this is his job for the next several months, is helping us turn these into commercially viable products that we can either sell or license, or actually kind of pair up with our core technology. And so yeah, so it's been a lot of fun.
So essentially a reagent that can directly deliver nutrients to all the sort of components of cells.
Yep.
Cell cultures.
That's right.
That's incredible.
Yeah, it's been really cool. And I can't believe that I came up with it. Honestly, if you ever make yeah, it's one of those things where we kind of just got lucky that it has all these amazing, we just came up with a cool approach. Like, I actually got frustrated reading the literature because people have used this for a long time, but it's like so arduous to like make it. And I'm like lazy. So this is really like came out of laziness. I'm like, what if we just did X, Y, and Z? And then sure enough, X, Y, and Z worked. And like, oh, and it's autoclave stable. Oh, and it's like very buffer stable. Oh, and it just keeps on surprising us with all these amazing properties. And if we just do like little tweaks, it can do all kinds of different things. So yeah, I'm really excited to explore that further. It is a lot of fun. Yeah, oftentimes I do feel like a mad scientist.
If you were sort of to give advice to young sort of scientists out there, what piece of advice would you give them?
I would tell them that they're already doing great just by being in science. Unfortunately, science is a very hard slog. They're going to watch their peers that are studying in other disciplines have Friday nights and Saturday nights off where they're going to be in the lab, where they're going to be studying. And it's going to be very painful sometimes. And I would just tell them to keep the broader picture in mind, because the reality is that if you have a science background, you're going to be more positioned to take on other disciplines in the future. Whether it's a podcast, whether it's a marketing specialty, whether it's writing. I think that having that scientific backbone is one of the most important things that you can do, because it gives you the analytical skills and so many other skills, to be honest. And it gives you like the resilience and when things don't go well, it gives you that exposure to the scientific method and experimentation and trying to push boundaries forward, where you get very used to working within spaces where you don't have all the information and be successful. So I would tell them not to get discouraged when they see some of their friends out maybe having more fun than them, and to stick with it, because it's very, very important. And you may have seen at the Built with Bio conference, Eric Schmidt's talk around the new bioeconomy. That it's a very great talk. I'm actually going to make a post on LinkedIn about it, because I was so moved. I thought it was just like one of the best talks I've ever seen. But his thesis is that working on biology is so for those that don't know, Eric Schmidt was the former CEO of Google. He's a Silicon Valley vet in every sense of the word. And so he's seen it all and done it all, and is very, 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 very bright. And his thesis is that the biorevolution that we're about to go into is going to be bigger than even the revolution that happened in Silicon Valley in the 90s and the 2000s and the 2010s. And not only that, but it has the opportunity to really be the driving force of economies, global economies moving forward. These big questions around climate change, these big questions around pandemics and how we deal with them, and that it's one of the best things that you can do for the world is to pursue science and also to commercialize your technologies. So not just keeping them to yourself, can you get them out into some broader context? So to bring it back to my previous point, where I want all scientists to become CEOs, I really mean that. And I would also encourage people to that are listening to this to read that report from the from the Schmidt group and take it to heart and see what white space is there and then pursue that. And I wish them the best of luck.